Intravascular amyloid white clot circulatory system deposition following primary particular viral infection and messenger ribonucleic acid vaccine administration. Has a novel pathology emerged? A literature review. Variegated post-mortem white clots, typical of those embalmers have encountered with increasing frequency in the COVID-19 era. Amyloid refers to an abnormal fibrous, single-stranded, insoluble, refrigerant, extracellular pathological protein deposition in organs and tissues possessing a beta sheet structure. A pathological cascade of intravascular amyloid white clot formation induced by cytokine storm and related dysfunction of the immune system, such as autoimmunity, has been observed in susceptible individuals. The white clots consist of amyloid, fibrin, and platelet components. Viral spike proteins projecting from the outer cell membranes that bind to host cell receptors, facilitating viral entry into the host cell, are believed to be amyloidogenic. These white clots are of a reported rubberish consistency as that of calamari, and of a configuration mimicking the structure and luminal dimensions of the arterial and venous vasculature, whereby blood seemingly flows in greatly reduced volume or not at all, and within sections of these thrombi. White clots have been observed in the course of medical testing, evaluation, and treatment in living patients, as well as upon post-mortem examination, and are logically believed to exclusively form in the living patient. A range of a dozen plus autoantibodies in some human beings directed against chemokines, cytokines, cell surface proteins, DNA, and RNA have been observed in the context of this formation of occlusive intravascular amyloid white clot pathology. Although 30 to 40 amyloid precursor proteins have been identified in a particular patient or disease, the pathological state is believed to be derived usually from a single amyloid protein. Energy-dependent functional three-dimensional protein misfolding is considered to be the main cause of amyloid aggregation and deposition, as well as a cause of various chronic diseases, since all bodily functions such as cellular division, enzymatic action, molecular transport, movement, nerve transmission, and secretion are affected by such conformational changes in folding proteins. Endothelial cells constitute the boundary between the blood and vascular walls in an energy-dependent manner, effectuating processes such as coagulation, fibrinolysis, thrombogenesis, vessel permeability, and smooth muscle contraction and elongation. Blood pumping and vascular endothelial dysfunction are believed to be capable of impairing organ perfusion, as well as perpetuating the prothrombotic state, resulting in various types of clots in the lumen of arterial and venous vessels. For example, human morbidity and mortality from strokes can result from, amongst other primary causes and or comorbidities, as well as white clot circulatory occlusion, as observed in some SARS-CoV-2 infected patients post-messenger RNA vaccination. These particular white clot formations were reportedly not previously observed in living and deceased patients prior to the advent of SARS-CoV-2 messenger RNA vaccination. References. Further determinative and confirmatory investigation, as well as peer review, is indicated concerning the emergence of a novel pathology corresponding to the research topic of this presentation. New presentations coming soon. Like, share, subscribe, and ring the bell for notifications. Thanks for watching. Thank you to current and new subscribers.